Hello everyone, let me introduce our group. We are from group 6. If we are from group 5 with the student name, first there is Annisa Firdaus with student number 1342000172. Second, there is Andrian with student number 1342000197. Third, uh, there is Alexis Ratri with student number 1342100214. For there is Hana Tsalisa with student number 134242000219. Five with there is Gabriel Stella with student number 1342000226. Six there is Muhammad Fafa Haika with student number 1342000238. Seven there is Sukma Melati with student number 1342200210. Our topic is about how plants defend from disease attacks. Next. In plant defense against pathogens, there are two aspects that need to be considered, namely, one, resistance, action or ability of plant to withstand or reduce the influx of a pathogens or other disturbance. Plants can free or eliminate an infection from a pathogen. Two, tolerant, action or ability of plants to accept infection or other disturbance without getting epidemic. Plants can still produce even though they get attacked. Next. Next, the opposite of resilience is vulnerability. Resilience and vulnerability are determined by measuring disease level, attack intensity, attack percentage, damage level, percentage of attack area.
Based on the level or intensity of attack, the plants are grouped into one, invulnerable, meaning there is no damage or loss at all. Two, highly resistant, meaning very little or almost no damage. Three, resistant, meaning little or no damage to the plant. Four, somewhat resistant, meaning that there has been loss or damage to the point. Next, five, somewhat sensitive or somewhat vulnerable, meaning that the damage or loss has been felt at their six, sensitive or vulnerable, meaning that there has been a lot of damage or loss suffered by the plant. Seven, very sensitive or very vulnerable, meaning that there has been tremendous damage or loss through the year or season, resulting in a complete failure of the health, level or resistance or susceptibility of plants to infection depends on two invariable factors, namely the need for food by the pathogen response to the pathogen. Plant defense mechanism against pathogen. One, mechanical defense. Two, chemical defense.
Okay, the next is mechanical defense. Mechanical defense of plants is usually by certain anatomical or morphological structure that can make it difficult for pathogen to make contact with plants, and ultimately the plant is protected from disease. These structures can be formed before penetration or after penetration by pathogen into plant disease. Next. And before penetration. Before penetration, also called passive defense, because there is already a certain structure of their plants before the pathogen attack the plants. This can happen if, first, the presence of thick layer of wax and cuticle on the surface of the plant's epidermis. The thick surface of the epidermis by wax and cuticle can prevent the plant surface from getting wet during the rainy season, so that it can make an obstacle from the attachment of spores of the plant surface. Finally, it can prevent some bacteria and fungi fungi from entering the plant tissue. Next one. And the second is accumulation of salicylic acid on the cell walls of the epidermis. Several studies have shown that the resistance of rice plant to blast disease caused by Pyricularia oryzae is related to presence of salicylic acid in the epidermal wall of rice plants. And the third is natural hole. Pseudomonas citrus, the cause of canker in orange, will only enter the stomata that are wide open. This is what causes mandarin orange to be more resistant than other type of orange. And the last is plant canopy. The location of the leaf has a significant effect to the efficiency of the inoculum attached to the leaf surface, and leaves located horizontally are more suitable for spore deposition.
Next, after penetration, also called active defense because the defense appears after the presence of pathogens that attack plants. Some of these defenses are the result of differentiation of tissue structure or disposition of tissue derived from tissue compound found around the area of attack by pathogen, which can also be called histological or histopathogenic defense. The first one is formation of cork layer. This layer can be an impenetrable barrier for pathogen. In addition, the layer can be penetrated by toxic compound released by pathogen. The second one, formation of abscission layer, formed to separate this tissue that is attacked by pathogen with healthy tissue. One to two cells that surround the pathogen will shrivel. The cell wall becomes thin and the pectin compound present in the middle lamella dissolve. With this layer and accompanied by the formation of synthetic substance and lignin that hardens this layer, the diseased plant cell will separate from the healthy plant cell.
The third one is tylosis formation in xylem tissue. Tylosis is an extraordinary growth of the protoplasm of living parenchyma cells to the side, which protrudes into the xylem space through spots. Tylosis has a cellulose wall that depends on many sizes so that it can clog the vessel involved. The tylosis can prevent the movement of pathogens, especially pathogens that attack vascular tissues. And the fourth one is formation of gum. In the attack of pathogens, sap is formed in the intercellular space and in cells around the area of infection, so that it becomes a fortress or barrier for vascular tissue pathogens, because the sap is a layer that is difficult for pathogens to penetrate. And chemical defense. Chemical defense divided into before penetration and after penetration.
Before penetration, the defense that occurs before penetration is also known as passive chemical defense. Compounds that can inhibit the growth of the pathogen before the pathogen arrives. So, the inhibitor is still formed at the third there is no pathogen. Most of the compounds found are phenolic compounds. From the result of this study, no less than 8 spots of phenolic compounds were found on paper chromatography. 4 of them were orthodeoxyphenols, which were higher in plant varieties that were resistant that that it is susceptible to workplace caused by curricularia origin. If the tissue is old, the inhibitory substance level are reduced, as it's resistant to pathogen infection. The next is after penetration. Plants can produce compounds that can inhibit the flow of pathogen and will only appear when plants are attacked by pathogen. It is also said to be active with plants. The compounds formed are called phytoalexins. Resistant and susceptible plants can both produce phytoalexins, but in resistant plants. In resistant plants, the amount of phytoalexin was produced in greater quantities and faster than susceptible plants. The mechanism by which phytoalexin inhibited the development of plant pathogen is currently largely unknown. Possible reaction that occur between pathogen and host plant cell membrane that have been damaged. In plant cells where pathogen enter, they generally experience rapid death, a condition of as a condition known as a hypersensitivity reaction.
Hypersensitivity reactions. Hypersensitivity reaction is a defense reaction in the form of rapid death of cell and surrounding cells that are attacked as a result of necrosis. The necrotic tissue can quickly isolate the pathogen and living tissue. As a result, the pathogen does not get nutrients, so it starves and dies. Hypersensitivity reactions occur only in unsuitable combination of host plants and pathogens. Changes, changes that occur in hypersensitivity reactions include loss of cell membrane permeability, increased respiration, accumulation and oxidation of phenolic compound, and production of phytoalexins. The end result of intensive mobilization of these internal reactions within the cell, invariably confirming and collapsing the infected cells and some surrounding cells. Pathogenic fungi and bacteria present at the site of the hypersensitivity reaction are isolated by necrotic tissue and die quickly. Other factors that cause resistance in plants. Number one, pathogens lack certain essential compound from the host, so that pathogens cannot inoculate, penetrate, or produce enzymes. Number two, in combination between the host and the pathogen usually produce a toxin that is specific to a particular host. Lack of host receptors for toxins produced by pathogens, so that toxin cannot be bound by host receptors, which makes the toxin not functional or unable to infect plants. Plants like this are called resistant. And the last one, lack of essential nutrients needed by pathogens, especially in obligate parasites.
Thank you. That's our presentation. If anyone has any question, they are welcome in the comments column.